Okay, hello everyone. Good evening, good evening, good evening. Good evening, good evening, good evening. Shivani, have you watched 11:30 class? No? Without fail, see, the moment you come from the college, watch the recorder and then only you attend this evening class, okay? Do not miss to watch the class of 11:30 a.m. Okay, perfect. Let's continue. So friends, in case of this, what we can say, in the cytokinin, we are just left with only one part of the cytokinin. That is what we left with, friends. We just left with the signaling process, is it? So we will finish that particular signaling process. By that, we will do the practice session. Good evening everyone. Good evening. I think very less people, no problem. Okay, so friends, let's continue. Okay, so I hope you all people remember in the morning class we have studied that cytokinin follows what kind of a component signaling system? Two component signaling system, is it? Everyone answer my question very quickly, friends. Apart from the cytokinin, everyone answer, apart from the cytokinin, what is the name of the hormone? What is the name of the hormone that is involved or that is using the two component signaling system? Everyone, what is the name of the hormone that is using the two component signaling system apart from the cytokinin? Everyone, what is that hormone? Of course, friends, ethylene. Correct, na? Good evening, everyone. Ethylene is the hormone which will follow the two component signaling system. Along with the ethylene, cytokinin also follow the two component signaling system. Wonderful, wonderful. Okay, so friends, now let me explain you the final step of the signaling. Okay, very good. And I hope every one of you remember my words very correctly. What did I tell to all of you? Friends, whenever you are studying the signaling, every student remember only 2 points. If you remember that 2 points, answering any kind of a signaling is very, very easy for us. What is that point I told to all of you, friends? You have to understand what happens in the plant body in the presence of hormone, right? And the second point, what happens in the plant body in the absence of the plant hormone? What happens in the plant body in the presence and what happens in the plant body in the absence? Is it, friends? If you people remember these 2 points, you can understand any kind of a signaling in a very easy manner. All the students follow my words very correctly, friends. Now look into the picture. Cytokinin, all the students follow my words. This is what kind of a domain, friends? Histidine kinase domain. All the students listen carefully. The moment cytokinin will come and attaches, where the cytokinin will come and attaches? Cytokinin will come and attaches to the extracellular domain. Friends, follow me. I will explain twice. Don't worry. Cytokinin will come and attaches to the extracellular domain. Friends, that extracellular domain of the cytokinin, what do you call it as, friends? You will be calling it as the chase domain. Friends, please don't get confused with this particular point. What do we call it as? Chase domain. Friends, understand my words very correctly. The moment cytokinin will come and associated with the chase domain, remember my words, it is going to transfer the information. I hope every one of you remember how does it will transfer the information. I have explained it to all of you very clearly, friends. It is going to transfer the information by the process of the phosphorylation. Yes or no, friends? Now all the students follow me. So look into this. Now it is transferring the information from histidine kinase domain to the receiver domain. But friends, all the students follow me. From the receiver domain where the information is passing. This is a very important point. From the receiver domain, the information is passing. Every student follow me. From the receiver domain, the information is exactly passing to the intermediate. What do we call it? This intermediate protein we can call the AHP protein. What does the A represents here? Arabidopsis histidine phosphotransfer protein. Now friends, understand my words. From the then AHP protein enter into the nucleus. Now every student has to understand. Now the AHP finally activating which domain, friends? AHP is finally activating the receiver domain. Friends, the moment AHP activate the receiver domain, what will happen? That receiver domain finally will show the output. Here you know what is exactly output means. It is going to activate some genes. What genes it is activating? Friends, it is activating the genes which are involved in the response of cytokinin. What do we call it? That response genes we can call the type A ARR respiratory genes and type B ARR. Friends, first of all, what is ARR? ARR means Arabidiosis response regulator. Understand my words correctly. What is called ARR? Arabidiosis response regulators. Now friends, what this particular AHP protein is doing? Now this particular AHP protein will come and activate the type A ARR, then followed by the activation of type B ARR also. But don't confuse my words. Actually friends, AHP first activate the type B ARR, then the type B ARR further activate the type A ARR. Or in some conditions what happens, you know, the AHP directly activating the type B ARR also, and AHP only directly activating the type A ARR also. So type B ARR and type A ARR ultimately going to show the response of the cytokinin. Friends, please do not get confused with that particular point also. Okay, now once again I am explaining the signaling pathway in just 1 minute, then we will move on to the questions. Cytokinin signaling pathway. Remember, so there is a, understand everyone, there is a presence of the cytokinin. Friends, there is a presence of the cytokinin. Cytokinin will come and associated with a chase domain. The moment it is associated with the chase domain is also called as extracellular domain. Immediately what is going to happen, friends? Histidine kinase domain get activated. The moment histidine kinase domain get activated, friends, understand my words, it will activate the receiver domain. And the point is very clear. How does the information is passing, friends? The information is passing, friends, in the form of a phosphate group. You can see the picture very clearly. In the form of a phosphate group, the information is passing. Now the receiver domain activating an intermediate molecule called as Arabidiosis histidine phosphotransfer. The moment Arabidiosis histidine phosphotransfer get phosphorylated, then through the nuclear pore it enter into the nucleus. The moment it enter into the nucleus, it is going to activate type B ARR genes and type A ARR genes. Now the type B ARR and type A ARR finally will show the response of cytokinin. This is the simple hormonal signaling of cytokinin. So friends, now we have studied all the 5 hormones signaling as well as 5 hormone structure, function, biosynthetic pathways. Now we are going to solve the questions. Now every student answer my question. If I solve 10 questions, if you are able to answer 6 to 7 questions by understanding the concept with the help of PPT, with the help of a lesson, if you are able to answer 6 to 7 questions, your preparation is going 100% smoothly. Your preparation is going very correctly. But if you are answering less than 6%, you have to increase your preparation level compulsory. Okay. Now, do not get hurry. Okay. Just be calm, be cool. Okay. Once again I am saying, friends, this line I don't mind to say repeatedly. 90% of the students, they're extremely intelligent, but when they are ready to answer the question, they will get excited very much because of the anxiety and because of overconfidence. Because of anxiety and overconfidence, they will end up with the choosing wrong answer, which means people are doing the wrong analysis. Okay, so that is the reason, my friends, whenever you are answering a question, you should not get excited at all. You should make the answer extremely simple. Okay, now let's start with the 2022 paper only, which is happened just a few months back. Okay, so this is as I told to all of you, friends, from the plant hormones we got 6 marks. Okay, now I want every student has to get that 6 marks. We are not reading also. We just listen the lesson, we study the PPT, we just did one revision. Now I will observe whether you can answer it correctly or not. Okay. Once again, don't take tension, slowly answer. Yes, start with the 4 marker question, part C. Take your time, then only you answer it. Don't be hurry. Trust my words, the statements are super confused. Slowly analyze it. I think Mauli, you have asked one statement in the morning class. The same statement only that was given in the question, you check it. Take your time, take 1 minute, no problem. Okay, super fast, Archana. Archana, make sure that you analyze the question very correctly. I don't want very fast answer, I want correct answer. Make sure that you analyze, everyone analyze the question correctly. Recheck your answers, but don't be don't be too confused. Make it very simple. Others, Deepika, Shafia, Mauli, everyone. Shivani, what will be the answer? Direct question from the PPT itself. Others, Karan, what will be the answer? Deepika, what will be the answer? Jahnavi, very straightforward, but little twisted. Archana said 4. Come on. Don't take too much of time and don't take very less time. Maintain a tempo. Just take 1 minute of time. Okay. Let me analyze all the options. All the students, please look into the options correctly. Shivani, what will be the answer? Jahnavi, Deepika, following are certain statements regarding gibberellic acid signal transduction. Look into the first point. Della proteins negatively regulate the gibberellic acid signaling. Absolutely true statement. Yes or no? Option A is true. There is no doubt about it. If you know that option A is true, you can eliminate the option 2 also. You can eliminate the option 3 also. Easy. Now option B says that degradation of GA receptor is mediated by the Della protein. Every student answer my question. Does the statement 2 is a correct statement, friends? Does the GA receptor can mediated by the Della protein? First of all, GA receptor never be degraded. Yes or no, friends? GA receptor never be degraded. GA receptor, they have given a statement that GA receptor degradation is mediated by the Della protein. Is that statement true? This is a wrong statement. At least if you analyze option B, you can easily come to a conclusion that the fourth one is the answer. Correct, na? Now look into so B is wrong. And what about the C? Ubiquitination and subsequent degradation of Della proteins are independent of GID1. No, they are exclusively dependent on GID1. Yes or no, friends? To the GGD1, who will come and attach us? Gibberellic acid will come and attach us. The moment gibberellic acid attaches to the GGD1, then the gibberellic acid signaling molecule will go and block the Della protein. Yes or no? It is purely dependent, but they have given an option that it is independent. So option B is wrong, option C is wrong. See, same thing. Gibberellic acid binding to the GD1 and promote the binding of GD1 to Della proteins, and then it will degrade. Obviously option D is right. Option A is correct, option D is correct. So the correct answer is 4. So 4 marks are in our hand. What difficult in this question? What you have to use your brain? Okay, very simple, very logical. Now what I can say this question? I can say that this question is a direct question. Okay, what you have to do is you have to analyze the options correctly, then you can choose the correct answer. It's not at all difficult. Okay, so you got 4 marks. Okay, now this is the part B question they have asked. And everyone know that. Very straightforward, very direct question. The plant hormone gibberellins are the group of every student. No need to think too much also. We all people know that. What do we call it, the gibberellin, friends? We call it the gibberellins as are the tetracycline. What do we call them? We call it them as the tetracycline diterpenes. Yes or no? Very straightforward question. So friends, just by reading the PPT, see, it is not about reading the PPT. What I am saying is with the help of concept, and just with the PPT, is there anything extra you have to read to answer these questions? You know, to be honest, you do not require the PPT also. If you know the concept correctly, 6 marks are in our hand. See, why I am stressing is I will prove now. I will prove entire September 2022 paper in all the units how easy the questions are. But overall I can use the word question paper is moderate. But trust my words blindly, you can answer 120 marks very easily by studying just PPTs itself. Okay, so I'll prove it because all the questions are very direct. Okay, now let's start. Next question, I think this is the 2018 question. The plant hormone gibberellic acid is generally not associated with. The plant hormone gibberellic acid is generally not associated with. Slowly, don't be hurry. Everyone, what is the answer? Tell me option A, B, C. Very good. You know, right? What is parthenogenesis? Everyone, I hope everyone remember. Spontaneous development of an embryo. Spontaneous development of embryo from unfertilized egg. What, friends? Spontaneous development of embryo from the unfertilized egg. That is what exactly we can call parthenogenesis. In the parthenogenesis, gibberellic acid never involved. It is involved in the stem elongation. It is involved in the parthenocarpy, and it is required for the malt production. Don't get confused with that, friends. It is absolutely true. So the correct answer will be C. Very good. Okay. This question also, trust me, you all people can answer. It is not at all difficult. Use the brain, apply the knowledge, take 1 minute time, analyze it correctly and tell me. I think someone has asked this question in the morning class, in the morning 11:30 class before I am leaving. I think Sumaya has asked about this particular statement. Someone asked, Sumaya or someone asked, ETR1 and CTR1. See, the same option, the same line was given in the question. You can easily analyze, friends, very easily. Take time, no problem, take time. Understand, Jagruti, try to answer. Krishna. Someone said option C. Archana answer C for what? Is this question or previous question? Okay, take time, take time, take time. I think it's previous question. Okay. Option C, huh? They have asked a question state which is correct. Friends, you are not listening my words, okay? You spend 1 minute. Don't come to the conclusion, please. Don't come to the conclusion. Don't be hurry, please. Take your time. Every student recheck your answers. Every student recheck your answers. Shafia said B, Karan said B, Mauli said B, Sumaya said B. Anyone else? Jahanavi? Shivani, what is your answer? Okay, now let's look into the statements. Okay, all the students follow my words very correctly. The question says that, okay, what is the question says? Everyone focus. Option D, okay. Ethylene is an important plant hormone that regulates several aspects of plant growth and development. Some statements are given below with regulation to the ethylene signaling pathway. I really, I can strongly say that the question is directly from the PPT. That's all. Directly from the signaling pathway. Okay, everyone listen. All the students answer my question. Option A says that unbound ethylene receptor work as a positive regulators of the response pathway. Now every student answer my question. Without ethylene, does the response can be shown in the plant? Everyone answer my question first of all. Option A. Without ethylene, does the response can occur? Everyone say yes or no to me. Without ethylene, does the response occur in the plant body or not? That is my question. Of course not. Without ethylene, there will be no response. Then you can easily eliminate the option A. Now done. Okay. Sumaya, please tell me. Yeah. Tell me everyone, Krishna, Deepika, tell me. In the morning class only we have discussed how many number of ethylene receptors that are discovered so far. There are more than 2 ethylene receptors. This statement is true or false statement. At least if you know this, you could answer the question. The direct line which I have written in the PPT. Yes or no? Karan, how many we discussed? There are more than 5. Yes or no? Which we can call the ETR1, ETR2 and ERS1, ERS2 and EIN4. Wonderful. How many we have studied? 5 we have studied. Now Archana, why did you give in the wrong answer in the first time? Then obviously option B is correct. Option B is correct. If at least if you all people know that option B is correct, you can come to a conclusion answer B. No doubt about it. Now I think Sumaya asked this statement in the morning. The carboxy terminal half of the ethylene receptor ETR1 contains a domain homologous to the histidine kinase catalytic domain. Yes or no, friends? Just now I have shown you the diagram also. This is the receptor, correct na? What does this receptor actually look like? It is looking like it can possess the activity of histidine kinase domain. In the morning class I have written example serine kinase, histidine kinase, RTKs. Obviously it will follow the C. So B is correct, C is correct. Okay, slowly. So B and C are absolutely true. Now what is this option D? Everyone please justify me option D, everyone. EIN2 encodes a transmembrane protein. The EIN2 mutation promotes the ethylene responses in both seedling and adult arabidopsis response. Everyone answer my question correctly. If the EIN2 got mutated, do you think ethylene response factors will be activated or inactivated? Obviously they will be inactivated. But what they are saying in the question, if there is a EIN2 mutation occur, the response will be shown. Never ever. If there is a EIN2 mutation, there will be no activation of the ERF. Hence friends, there will be no response only. Yes or no? Of course, true point. So this option D is wrong. So option B is correct, option C is correct, option A is wrong. Hence the correct answer will be option B. Shivani, Jahnavi, is it clear? Understand correctly or not? See, 4-marker question. Okay, everything how you nicely analyze the question. If you are fail in analyze the question, you can never answer the question. Always analyze the question properly. Wonderful, wonderful. Okay, next question. Dark grown seedlings display the triple response when exposed to the ethylene. Which one of the following is not a part of the triple response? Which one of the following is a part of the triple response? Which one of the following is the part of the triple response? Morning class also I have told to all of you. I have shown you one diagram, right? Okay. Anand, you have missed few questions, right? Without fail, watch the recorded, Anand, okay? Whatever the questions that you are not there, watch recorded without fail. Okay. Dark grown seedlings. We can also call it as what seedlings, friends? Etiolated seedlings. They are going to display the triple response when exposed to the ethylene. Which one of the following is not a part of the triple response? Okay, so we discussed decrease in the epicotyl elongation, nothing but inhibition of the epicotyl elongation. This is right. So you know, right, friends, does ethylene having any kind of a role in the expansion of the leaf, rapid unfolding? Okay, now every student please answer my question. Every student, what is the hormone that is involved in the rapid unfolding and expansion of leaves? Morning class only I told you. What is the name of the hormone? Karan, can you please tell me? What's the name of the hormone that is involved in the expansion of leaf? Of course, friends, cytokinin. Correct. Now I have shown you the experiment also. Cytokinin is the one which is involved in the leaf expansion. Correct. Now, and thickening of the shoot also it will only do horizontal growth of the epicotyl. So which is not a part of the triple response? Obviously, option B. Rapid unfolding and expansion of the leaf. Wonderful. Very good. This is the part C. This is very good, friends, very good. This is the part B question. And what is this? This will be the part C question. Okay, I am telling every student take 1 minute time and remember my words. Every student has to answer this question. Okay, in the morning class when I am saying something someone has asked, then I have explained 1 minute about transmembrane receptor and soluble receptor. Don't be hurry, use your brain and answer my question. Four-marker question it is. (2019) paper. Take your time. Wonderful, wonderful. See here, how easy question which we got in a 4-marker. The question is easy if they have asked differently. Auxin and gibberellins. Correct na? They are having what kind of receptor? Soluble receptor. Very, very good. Correct now friends? And what is the function? We already discussed. The moment auxin will come and attaches to its receptor, what it will do? It will inhibit the inhibitor by the proteasomal degradation. So AY2, and friends, everyone remember, cytokine and brasinosteroids will do the X and 1. Correct na? Response mediated by receptor phosphorylation or the dephosphorylation. Absolutely true point. There is no doubt about it. Hence friends, this will be BX1 and this will be the AY2. Hence the correct answer will be B. Wonderful, wonderful. So everyone given a very correct answer. 4 marks are in our hand. Simple it is. Okay. Next. Which one of the following amino acid is the characteristic of and it is converted into a response regulator of plant 2 component signaling system? Yes. Shafia, I think you forgot, no? See, we discussed, no, right? Which contain 2 component signaling system? Hormone ethylene contain the 2 component signaling system. Hormone cytokine also contain. Both are having what kind of domain? Histidine kinase domain. Correct? Now, it's not an aspartic acid. So, the domain contain basically full of amino acids which are histidine, right? Hence, the correct answer will be the histidine. So, I solved the questions from the 2022 paper to the 2017 paper. Okay, now just everyone respond to me. Is it clear everyone about the all 5 hormones correctly or not? And I hope all the students has given the correct answers for every question. I hope you got a confidence that you can solve any kind of a question that is asking from the hormones. Yes or no? Aspartate is also involved. There is no doubt about it. Aspartate is also involved. Aspartate's function is what? It is involved in the process of phosphorylation. Correct na? It is involved in the process of phosphorylation. Absolutely true. But information is going from where? Information is going from the histidine domain only, right? From the histidine domain only the information will start in the form of a phosphorylation. Then obviously histidine will be the major amino acid. Right, then aspartic acid. Okay, I hope everyone got it. Wonderful, wonderful, wonderful. Okay, so friends, what is the tomorrow topic? Tomorrow topic is the photosynthesis. So because you know it, a very big topic, and if you look into this 2022 September paper, we got part only part A question. We got a part A question from the September paper, only part B question, that is for the 2 marker. This time I am believing 99.99% whatever the exam that you are going to write in the photosynthesis, you will get a very clearly the 4 marker question itself, as well as you can get, if there is a great chance you can get the 4+2 also. So in the 5:45 AM class and 11:30 AM class, because it's a very big concept, I'll finish the entire photosynthesis. In the evening class, we will solve the PYQs. Okay, that is the goal. Okay, now the time is 7:35. I am giving 15 minutes of time right now. All the students read 5 hormones once, and any kind of a doubt you ask me right now, I will explain it right now. Okay, so I am also studying along with you 5 hormones. 15 to 20 minutes I will study all, then we will end up the class. Any kind of a doubt ask me, I will resolve instantly. Once again, we don't touch the hormone. You people has to do revision, not now, after few days of time. Okay, now let's revise it once again. I am giving 15 to 20 minutes of time. Everyone start studying it. Please don't leave the class. Study sincerely. Okay. Please don't leave the class. Study sincerely and ask me any kind of a doubt. By sharp 7:55 I will end it. You can ask me any question. Start study. Please don't leave, read 15 to 20 minutes sincerely. By that we should not touch the topic again. Majorly focus on the precursor signaling functions. What Archana, you have asked a question. Please explain under physiological effects of a gibberellic acid fruit set point. Okay. Archana, please look into this. Archana, follow me. Okay. Gibberellic acid will be involved in the every student whomever did not understand that point, listen. Gibberellic acid is involved in the internodal elongation. Not only internodal elongation, it will be involved in the axis elongation also. Okay. Now remember, okay, suppose we all people know that how does the cabbage look like. Okay, listen here. This is the cabbage. Arey yaar, I don't know why it is creating irritation. Okay, 1 second. 1 second, wait. I'll take another slide, by that it comes nicely. Perfect. Okay. Archana, look into this. You know, right? Actually, the axis of the cabbage is very small. Axis of the cabbage is very small, and you know, all the cabbage leaves will be accumulated like this. The axis is very small and all the cabbage leaves will be accumulated like this. Okay. Now, when I injected a gibberellic acid, the middle axis will increase. The middle axis will increase. The movement axis will increase. What happen? All the leaves of that particular cabbage can freely arranged. As a result, what is happening? This particular cabbage looks very bigger. Not only cabbage, Archana, remember grape. Suppose example, this is the axis of a grape. So if you look into the grape, see, axis is very small. To this particular axis, abundant amount of grapes will be associated. You can see here. You know, sometimes what happen, because of a, remember my words, because of a small axis, grapes will fall down. Because of the small axis, grapes will fall down. So that time what we do, upon injection of the gibberellic acid, the axis will be increased. Once the axis will be increased, all the grapes can easily accommodate. I'm sorry with this writing. Once the axis will increased, you know, grapes will be automatically accommodated easily. This is called as what? Fruit setting. Even let's take an example of a apple. You know, right? Originally the apple will be this much size. This is the size of an apple, small apple. You know, some people will say that the apples are purely came from the Kashmir. If you look into the Jammu Kashmir apples, there the size of apple is very high. Naturally, the apple size is extremely high and thick, very thick. But if you look into the apples in other areas means what? Where the temperature is very high. Let's take, if you take other north side, where the temperature is high, this particular apples are not great in size. Now what the people will do, once again upon injecting this particular gibberellic acid, what happens? Here the width of the apple is increasing. So basically why we will inject this particular gibberellic acid is what? Commercial purpose. Okay, so in case of apple, the gibberellic acid is increased the width. In case of a cabbage and grape, it increased the axis. So this is called as actually axis elongate. Sorry, this is called as what? Generally fruit setting. Which means if there is an increase in the size, obviously more fruits can be accommodated. Obviously it is commercially highly useful. Understand very correctly. Of course, auxin also will do the same function, but when compared to the auxin, gibberellic acid is the one will plays the crucial role. Auxin cannot do the same functions in all plants. Auxin is also involved in the fruit development. There is no doubt about it. Because auxin is involved in the parthenocarpy, right? It is also involved in the fruit development, but not in all the cases. But gibberellic acid will do the active role in the fruit setting. Got it correctly? Ask me once again if anyone did not understand. I'll re-explain with another examples. So gibberellic acid induces the internodal elongation and axis elongation. By that, it will create a more space for the leaf and fruit to accommodate easily. That is what exactly we can call it as a fruit setting. I hope everyone remember, right? Gibberellic acid is the one which block the seed dormancy. If it block the seed dormancy, obviously the fertilization occur, seed germination occur, size will be increases. There is no doubt about it. Simple point it is. Okay, 10 minutes more. Please read and ask me any kind of a doubts, any kind. Okay, you want ABA signaling, Mauli? Okay. Mauli, quickly look into the signaling of ABA. ABA, or what is you are asking? In stomatal guard cell opening and closing. Okay. Okay. Look into this. Okay. Look into this, Mauli. This is called as guard cell. Archana, I am coming. See, every student remember, ABSCISIC acid, see these two are the guard cells. On guard cell, what kind of receptor is there? The receptor for the ABSCISIC acid, correct na? Now, the moment ABSCISIC acid will come and attaches, ABSCISIC acid immediately activating two pathways, ROS pathway and IP3 cyclic ADPR pathway. Every student remember, ultimate goal of the both pathways are what? Sending potassium outside, sending chlorine outside, sending water outside, and bringing what inside? Calcium inside. This is the ultimate goal. When we send all these out, it will become completely flaccid. Then the flaccid cell easily closes the stomata. That's the original concept. Now all the students follow me in the ROS pathway. When abscisic acid associated with the abscisic acid receptor, abscisic acid activate the ROS species. Example H2O2 O2 minus. Now all the students follow me. This ROS species, remember Mauli, ROS species will perform three functions. What are the three functions performed by the ROS? It is opening the calcium channels. By that the calcium comes inside. The moment calcium comes inside, it blocks the potassium incoming channel. No potassium comes inside and it will allow the potassium chlorine goes out. So three functions are what? Bringing calcium inside, blocking potassium incoming, sending potassium chlorine outside. When the potassium chlorine goes outside, obviously what happens? The guard cell will become flaccid. Then the flaccid guard cell close the stomata. This is ROS pathway. Along with the ROS pathway, simultaneously IP3 cyclic ADP ribosylated pathway also occur. What is happening here? Whenever abscisic acid will come and associated with the abscisic acid receptor, what is happening? This abscisic acid will activate the phospholipase C. We called it as the PLC. Now the PLC activate the IP3 molecule. And what is the function of an IP3 molecule? IP3 is a secondary messenger. It brings the calcium. Now where the calcium entered? Calcium entered into the cytosol. The moment calcium comes, potassium channels, potassium goes out and proton pumps also goes out. I mean to say proton pump activated, proton goes out. Now H plus gone out, potassium also gone out. What is happening now? The cell becoming turgid. The cell becoming flaccid. Because of the flaccidity, what is happened? Again the stomata is getting closed. Whether it is the ROS pathway, whether it is the IP3 pathway, friends, the point is very common. They are sending the potassium chlorine out by as well. Generally, they are making the guard cell to become flaccid. As a result, they will close the stomata very, very easily. Don't get confused. But the point is here, both pathways are occurring simultaneously. Don't get confused. I hope it's clear for everyone. You understand, Mauli? What is your question, Archana? Archana asked a question. Is there any inhibitor for the control or regulation of abscisic acid biosynthetic pathway? Actually, remember, it's not about the pathway, Archana, but remember my words very clearly. Abscisic acid actually controlled by a process called as oxidation. Oxidation or conjugation. Okay. This is the process which is actually acting as a regulatory mechanism to control the abscisic acid. You have to understand, sir, what happens here? Your question is really valid question. Oxidation, in the process of oxidation or conjugation, abscisic acid actually can be converted into 6-hydroxymethyl ABA. 6-hydroxymethyl ABA. And 6-hydroxymethyl ABA is completely inactivated ABA. So when the ABA level becoming higher, what happen? Oxidation or conjugation process will be activated. They convert the ABA into 6-hydroxymethyl ABA. Then the ABA will get inactivated. Got it? The mechanism is oxidation or conjugation. No. Proton pump is not at all inhibited by the calcium, Shafia. That is wrong statement. Look into the picture. Calcium is actually stimulating the proton pump. You're understanding? As a result, what happened? All the protons completely goes out and protons never come back. So proton goes out, potassium also goes out. All the positive charge goes out. Don't get confused. As a result, what is happening? Obviously, the flaccidity is getting increased in the cell. As a result, stomata closes. Okay. Okay, I hope all the doubts are clear. Okay, so I am requesting all of you right now only. Okay, please don't take too much of time. please take 15 to 20 minutes and revise all the hormones once again. So make sure that how difficult question you may get in the exam, you should be in a position to answer that particular question. Okay, I will meet tomorrow 5:45 a.m. sharp. Our tomorrow ultimate goal is finishing the concept of photosynthesis and solving the PYQs. Okay. Okay friends, that's all for now. I will see you tomorrow sharp 5:45 a.m. Okay. Thank you all of you. Thank you so much. Okay. Thank you. Thank you.